Thank you for the kind introduction. Good evening. Firstly, I want to thank the Royal Family of Sweden for their hospitality, the Nobel Prize Foundation for the honor, and honorable guests of Sweden for their presence. The topic of my presentation is adeno-associated virus in human gene therapy. I will try to make the presentation as simple as possible to cater to the diverse audience. On the top left is a small picture of adeno-associated virus, AAV. This virus is a small group 2 non-replicative, non-enveloped, single-stranded DNA virus belonging to the family Parvoviridae, genus Dependovirus. It infects humans and some other primate species. AAV is not currently known to cause disease, and consequently the virus causes a very mild immune response. Gene therapy vectors using AAV can infect both dividing and non-dividing cells and persist in an extrachromosomal state without integrating into the genome of the host cell. These features make AAV a very attractive candidate for creating viral vectors for human gene therapy. This slide shows entry of AAV into cell. After delivery into the human body, AAV first evades the human immune system and antibodies and reaches the target cell. By receptor binding, then AAV enters the cell's cytoplasm through the endosome and is transported by late endosome up to the nuclear membrane. It escapes from the late endosome and cellular degradation, proteasome, and reaches the nuclear membrane. By transduction, AAV enters the cell nucleus, the genetic material is released single-stranded, and the genetic material is converted to double-stranded nature by cellular machinery. Some double-stranded AAV exists in cell nucleus as circular episomes, while the majority of AAV integrates by fusing itself at a highly specific site on human chromosome 19 by forming a concatemer. This slide is a detailed slide showing the behavior of AAV once it enters the cell nucleus, once again illustrating AAV's integration onto a highly specific site on human chromosome 19. This is a genetic locus map of human chromosome 19, which indicates all the human diseases and human traits with which human chromosome 19 is associated. Once AAV/AAV2 reaches the cell nucleus, it can go to one of two pathways. In the absence of a helper virus, AAV/AAV2 integrates as mentioned above on a specific site on human chromosome 19 via the latency pathway. In the presence of a helper virus such as adenovirus, AAV follows the productive pathway by replicating and forming many small AAV virion particles. The point here to understand is that AAV by itself, in the absence of helper virus, is a replication incompetent virus. When AAV is delivered to the human body, we are not capitalizing on delivering a small amount of AAV and hoping for it to replicate and grow within the body, but rather we are delivering a high concentration/adequate dosage of virus, which will eventually follow the latency pathway. This slide displays the behavior of AAV in the cell nucleus. As mentioned earlier, some double-stranded AAV exists in cell nucleus as circular episomes, while the majority of AAV integrates by fusing itself at a highly specific site on human chromosome 19 by forming a concatemer. Here is a slide showing the site on human chromosome 19 viz. AAVS1 or Q13.3-QTER onto which AAV integrates itself. AAV/AAV2 genome map. Understanding this is important.